So the first thing we're going to do to be able to make that connection between meiosis and genetics is we're going to do just a really quick meiosis overview. So that's where we're going to start with this. And so with meiosis, the main takeaway is that we started with a diploid parent sex cell. So that's what we began with in meiosis, was a diploid parent sex cell. Remember, sex cells, collectively called gametes, are testes in males and ovaries in the females. And then interphase occurred, not shown here. And then after interphase, when the DNA and certain key organelles were replicated, then we have our first round of division known as meiosis 1, which consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. After that first division, you have haploid cells. And it's not because of the chromosome number, it's because of the amount of genetic material. So after that first division, even though the chromosomes have duplicated and homologous pairs have separated, you still only have half of the original genetic material. Any way you look at it, you only have half. And then after that first division, we have a second round of meiotic divisions known as meiosis 2, which consists of prophase 2 and metaphase 2 and anaphase 2 and telophase 2. There is no interphase before that second round of meiotic division because our goal is to reduce the chromosome number by half, and we can only do that if we have one round of replication and two rounds of division. So that's your meiosis overview. And then the end result here is we get those four haploid, and this is the important part, unique, they're all different, gametes. And so when we talk about gametes, if this was a testes cell that was going through meiosis, we get sperm. And if it's an ovary cell, then that's where we get an egg and those three polar bodies that we discussed. So let's take a look at this for a second as far as the relationship that meiosis has with a Punnett square. And so I'm going to draw out a Punnett square like you've typically seen before. But instead of beginning by throwing letters all over the place and using those letters on the outside that you may not even know what those letters mean yet, we're going to do something a little different. And we're going to say that on the outside of this Punnett square, across the top, we'll say our sperm. And then along the side here would be our egg cells. Now, here's what you probably didn't learn. Whenever we work out a Punnett square, what we are representing is the act of sexual reproduction, where each square results in a fertilization event, or the probability of one, I should say. And so within each square, that is a potential zygote. And I say that it's a potential zygote because one of the questions that we ask sometimes is we'll look at a diagram like this and ask, if a couple had children, if this male and this female had children, how many children would they have? And typically the students will answer, well, they have 4 children. And that's not the case. They only have one at a time, but within each child, they have these 4 different possibilities. And so that's really what a Punnett square is it's looking at the possibilities of a male and a female going through sexual reproduction to produce a zygote. So what we're really looking at when we do Punnett squares is we're looking at numbers to make predictions of what we expect to see in the offspring. And so if we look at that on a large enough scale, then we're looking at a whole population. And that's where those of you that have experienced chi-squared and calc or stat, you are familiar with the observed and the expected numbers. So we are comparing what we actually observe to what we expected to observe. And what that does for us is that allows us to determine whether or not a population is changing or evolving. And so that's going to tie in to what we look at when we get to evolution later on. Okay, so I'm going to block this off. And again, we're going to deal with just a regular 4-member Punnett right now. And we can look at this a couple different ways. We can say that each square is 1/4, right? 1/4, 1 out of 4. Or we can say that each square is 25%. And so our goal when we're reading these is to make sure that our predicted outcomes either add back to 4 or add up to 100%, depending on if we're dealing with ratios or probabilities. Okay, so we're going to look at the different ratios that we might see with our typical 4-member Punnett when working out what we call monohybrid crosses. And we'll define that later on, but just know that monohybrid means we're looking at one trait and one trait alone for the time being. So I'm going to make 3 columns here. And in one of those columns, I'm going to call that my Punnett square. And I'm going to use some color to differentiate with this. So if you're writing this down, I would recommend you use some color as well, whether it be colored pencils or colored pens, so that you can differentiate the way that I am going to. And then I'm going to give you a ratio, a possible ratio, and then I'm going to give you a probability. And so what we've got to be able to do is get comfortable enough between these so that we can go back and forth, because they're all saying the same thing, but we're right back to the concept in life science where we have different models that represent the same thing. So this first Punnett square that I'm going to draw, I'm going to leave it blank. And I'm going to leave it blank on purpose because we're going to pretend that the question has asked us for something that we're not seeing. So it's asked us, how many would you predict that would have trait A, whatever trait A may be? And so we're going to pretend right now that there are none that we would expect to exhibit trait A. And so that would be my ratio, as I would put, none. Probability would be 0%. Now, if my Punnett square were to contain, and again, I'm going to use colors for traits right now, all of the same thing, and so that's what we're looking for. We've got exactly what we expected to see. Then on the flip side of none, we would say that we would predict them all to be blue if we're looking at color. And the probability for that would be 100%. Now, in this next Punnett square, I'm going to make 2 of them blue, and I'm going to make 2 of them pink. Now, technically, this is a 2-to-2 ratio. However, and you guys have learned this in math, we don't leave it at 2-to-2. We reduce it to a 1-to-1. And so this would be a 1-to-1 ratio. And the probability of that would be 50% to 50%. Another scenario that you might see is you might see 3 of these that are the same, and one of them that's different. So if you see that scenario, that would be a 3-to-1 ratio, and the probability that that would translate into would be a 75% to a 25% ratio. One other example, and we won't see this as much, we'll never see it phenotypically with a standard dominance problem, but we'll see this with incomplete dominance. We'll see this with codominance. We will see examples where we will have 3 different things inside of the punnet. And so it will look something like this. And the ratio for that will be a 1 to 2 to 1. And then that translates into a probability value of a 25% to a 50% to a 25% probability value. So let's tie this back into chromosome structure that you guys learned about. And so I'm going to clear this screen to free up some room for us. And so some terminology, we're going to take a look at some terms that you guys are going to hopefully be reminded of. But if it's been a while, if you're a senior and it's been a freshman since you have seen this, then these terms are really important. So without understanding those, you need to be able to take a word problem and convert it into a genetics problem. And so one of those terms that you need to be familiar with are these things called alleles. And we're going to spend just a minute with this and relate this all the way back to organic molecules, which I know for some of you kind of makes you cringe because that was not your favorite unit. But I'm going to give you a very non-textbook definition of alleles. And what we're going to call this is we're going to call these alternate forms of a gene. Now, we defined gene way back when, but we can simplify that to be a segment of DNA that codes for protein. Okay, so if you remember back in organic molecules, one of the things that we learned was that the order and quantity, or the sequence and number of nucleotides in a gene, are what ultimately determine the order and quantity of amino acids in a polypeptide. And that directly translates into primary structure, which is the most important type of protein structure because it literally dictates everything from there on. It dictates secondary, tertiary, quaternary structure, and that shape determines a physical trait or physical characteristic. So alleles are alternate forms of a gene. Now, for alleles, we use letters to represent them. And we're gonna deal with flower color, one of the traits that Mendel observed in pea plants. And so we're gonna say that a big P is going to represent purple, and we'll say that a little p is going to represent white. So since this is AP biology, you actually have to know what those letters mean. And so we're going to tie this back into those homologous pairs that we learned about in meiosis. And I'm going to keep the same color scheme that we use there. We'll say that this is, we're going to make chromosome pair number 1. And so with chromosome pair number 1, we're going to say that this red chromosome represents the maternal chromosome. And we'll say that the blue represents the paternal chromosome. And we'll just say, and don't quote me on this because I'm not sure exactly which pea plant chromosome pair codes for flower color, but for argument's sake, we'll just say that this is homologous chromosome pair number 1. We'll say that the maternal chromosome of this pea plant has big P, and we'll say that the paternal chromosome has little p, okay? So here's what I want you to understand is exactly what those letters known as alleles mean. So that P is representing a strand of that maternal chromosome's DNA, okay? And that DNA is what we're going to call a gene, and we'll call the big P the dominant allele. The little p we'll call the recessive allele. And what that little p is doing is representing a segment of DNA on the paternal chromosome, which is also a gene. If we look back up at the top, by definition, what a gene is a segment of DNA that ultimately codes for protein with one really important step along the way, and that it first codes for messenger RNA. So when we get to our next unit and learn the intricacies of DNA replication and also protein synthesis, that's where we'll begin to focus more on the RNA. But ultimately, what that RNA codes for is a protein that makes the flower purple. And the same thing with the little p, that is an allele or a letter that's representing a gene that codes for mRNA that makes a protein that is going to make that flower white. Okay? Now, another thing that we need to be sure we cover before we just jump right into working out all these different types of Punnett squares is we need to be sure that you understand the steps required to work out a Punnett square. And you may be thinking, well, Charles, we know how to work out a Punnett square. We've done those before. If you ask most people what the first, how would you start? What's the first step in working out a genetics problem? They would say, oh, you got to do a Punnett square. And that may be true if it's a very, very simple problem, but we're not going to be working through very many simple problems. And so there's a series of steps that you need to understand and some more terminology that you need to know before we jump into some practice on these types of problems later this week. So we'll title this Steps to Solve Genetics Problems. And so these are steps that you would want to take, especially the first few. These are steps you would want to take to solve a complete dominance, incomplete dominance, codominance, blood typing, sex-linked traits. These are steps to be able to read a pedigree. These steps will allow you to, and the good thing about it is one step leads to the next. So you get the information from one step, in order to be able to go on to the next step. So they are synchronous, and if you follow these, then the probability of you getting a genetics problem incorrect greatly decreases. So the first thing that you've got to do is called the allele assignment. And this is something that you probably have not had to do before, because in your honors biology class, you were probably given, hey, use big P for purple and little p for white. So it may not give you that, and it may not even tell you which one is dominant. It may be a word problem, and I'm going to show you some examples of those where you're going to have to figure out which one's the dominant one and which one's the recessive one. So the first thing you got to do, and we'll keep with our flower color here, is you've got to do your allele assignment. You've got to make sure you include those letters. My advice to you when you're starting out doing these allele assignments is that you figure out whatever the dominant trait is, and you use a capital letter. So like purple, use a capital P for that. So you use the letter of the capital letter of the trait description in order to do your allele assignment. The second thing you need to do is a test cross. And this gets left out a lot. And if you leave out the test cross, then you stand a chance. And again, we're looking at more simple problems first. But when we start getting into these more complex genetics problems, if you leave out the test cross, you run the risk of not setting up your Punnett square the right way. And if you don't set up your Punnett square the right way, then you're not going to get your correct ratios and probabilities that we looked at earlier. So in this case, I'm going to say that, and we're going to call this the P-gen. And this is the one we're starting out with. So the P-gen stands for the parental generation or the parent generation. So this is where we're starting, and we'll just say that we're going to cross what we're going to learn is a homozygous dominant purple plant with a homozygous recessive white plant. So there is our test cross. The third thing that you want to do is your Punnett square. So now we're to the point where you can set up that Punnett square. So we used our allele assignment to set up our test cross. We're using our test cross to set up our Punnett square. So what you do is you take one parent, and we'll say this is going to be our first parent, this big P, and you separate the alleles over the top of the boxes. Now, here's what's actually occurring when those two things separate. When they separate out, we call that the law of segregation. That's Mendel's law of segregation. Where that occurs is in anaphase 1 of meiosis 1 and anaphase 2 of meiosis 2. So whenever we separate those homologous chromosomes and then ultimately separate those sister chromatids, that's what we're doing, is we're separating those through the process of segregation, where they get packaged into their own individual gametes. And so the other parent is separated through segregation, or what we call disjunction, in the anaphases. And then whatever's on the top, we drop down into the bottom. And whatever's on the side, we bring over. Now, if you run into a situation like we did here, where there are boxes in your Punnett that have both a dominant allele and a recessive allele, you always want to make sure that you put the dominant allele before the recessive. So you don't want to put a big P, little p, and then a little p, big P. It gets confusing. Now, it's going to be a little different when we look at incomplete and codominant problems, because we're going to be using different letters, and both of those letters are going to be capitalized. And so I'll give you some strategies there to help you keep things in alignment so that you don't flip-flop and mix them up so that it looks like different things when it's actually not. So we'll cross that bridge when we get there. The next thing that you do, and here's where some terminology comes in that you've either not seen before, or if you have, it's been a while. The next thing you're going to do is you are going to get your genotypic ratio or your genotypic probability. Now, a couple of terms that I want to give you, obviously they're all the same, so this is going to be a very simple ratio here. But what you want to do with this genotypic ratio is you got to understand the terminology that goes along with genotypes. So there are some terms here that you need to understand, and these terms are what's going to allow you to read a genetics word problem and convert that into an actual workable problem using these 5 steps that I'm showing you now. So genotypes can either be homozygous or heterozygous. And we're going to look at the homozygous examples first. So homozygous genotypes would either be big P. Again, homos means same. We learned in meiosis, zygous means joined, so same joined. Big P, or little p. Okay? So big P or little p would be a homozygous genotype. Now, there's some other terms that go along with homozygous. You might hear this referred to as true breeding. You may also hear a homozygous genotype referred to as purebred, or they may simply call it recessive, okay? And the reason why we can call it recessive is because the only way something can be recessive is if it's homozygous recessive. So those terms, and we're right back to the old saying in biology where we have different names that mean the same thing. If you hear something referred to as true breeding, purebred, or recessive, that means it's homozygous. The other option for genotypes are heterozygous, and there's only one example for that in this case, which would be big P, little p. Heteros means different, zygous still means joined. And some other words that you might see with heterozygous, sometimes they'll call it a hybrid. Sometimes it's referred to as mixed, or when we get to sex-linked traits, you'll hear heterozygous individuals referred to as carriers, also with some autosomal traits. So if we look at our genotype, which we get directly from our Punnett square for this example, they were all big P, little p. So we would either say all big P, little p, or we could say 100% big P, little p, depending on if the question was asking us for the ratio or the probability. Last step. Step 5. Phenotypic ratio. Here I'm going to give you an alphabet analogy. So for phenotypic ratio, we are looking at the physical characteristic. So whenever we talk about phenotype, we're talking about what we can actually see. So we can't see the genotype. Those are the letters known as alleles that represent the DNA that's coding for a certain protein. And what we're actually seeing is what that protein results in. So that's the phenotype. So in this case, where we had all of the same genotype, we will also have all of the same phenotype. And in this case, that would be all purple, because big P, little p, the purple masks the white, or 100% purple if it's asking for the probability instead of the genotypic ratio.